Next challenge was to get the agonist of erythropoietin receptor by using Fuzzy display. Erythropoietin made under low oxygen tension from the proximal tubular of kidney stimulate the stem cells in bone marrow, inducing the production of the red blood cell by some complicated steps, compensating the oxygen deficiency. The mechanism how EPO stimulate the stem cells in bone marrow starts with binding to the EPO receptor. As you see here, binding just a change, conformational change induce the series of reactions occurring in cytoplasm, ultimately leading to the gene expression. So this EPO molecule is kind of large, but basically in this project they try to make a small size of peptide mimicking the EPO. EPO erythropoietin has 165 residues shown here, and they just try to make a peptide which is 20 residue long, called EMP1, functioning as agonist of erythropoietin receptor. So receptor, the EPOR, has an extra domain. For this study, they just used its extra domain called EBP. They are successful in obtaining this 20 residue long dimeric form mimicking EPO. We're going to see how they achieve this goal. EMP1 was selected by 2 cycles of Fuzzy display. First, they just use the sequence shown here, where the center 8 residues were randomized, and also ending with 2 cysteines at both ends. And next step, they just extended the length of a peptide, something like this. So here again, they just partially randomized the center residues, and also together the two both ends. 20 residue long EMP1 they had obtained shows KD value of 0.2 micromole and shown to be stimulating EPOR activity in vivo. You can see the structure of EMP1 dimer. In the presence of EBP, this EMP1 peptide forms a dimer. Each monomer, shown in red and blue, forms beta hairpin structure stabilized by hydrogen bond, red dashes, and the disulfide bond, the yellow. 2 peptides form a symmetrical dimer stabilized by 4 hydrogen bond and hydrophobic contact. 2 monomers form a 4-stranded anti-parallel protein sheet. Binding of EMP1 causes dimerization of extracellular domain of erythropoietin receptor, shown here. 3 residues thought to be critical for binding erythropoietin, phenylalanine 93, F205, M150 are fully buried in the structure of peptide and receptor complex. Many biological processes are activated by hormone receptor interaction, and a great deal of research has been devoted to identifying peptides or small molecules which either inhibit or stimulating the hormone function. The idea that a small molecule can mimic for the function of a large protein is based on the hotspot principle, that a small molecule of residues at binding interface contribute most of the binding energy. The EMP1 selected by fuzzy display from random peptide library demonstrate that a dimer of 20 residue peptide can mimic the function of monomeric 166 residue protein, even though they are different in sequences. Thus, there can be a number of ways to solve molecular recognition problem. Natural selection works through the complementary processes of mutation and genetic assortment by recombination. Comparable in vitro experiment is going to be DNA shuffling. As you see here, two processes, mutation and recombination, occur together. You can see the details in previous slide. DNA shuffling about the cephalosporinase gene. Its gene product is involved in bacteria resistant to some antibiotics called moxilactam. In A, 4 homologous genes from bacteria were subjected to one cycle of cleavage and recombination, resulting in mutant genes containing 4 mutations, shown in red circles, and large pieces of each wild-type sequence. These mutant genes were transformed into bacteria and screened for resistance to the antibiotics. The most active mutant increased resistance by 540-fold over the wild-type genes. By contrast, DNA shuffling of each individual gene yielded only point mutations. The most active of these mutants only increased resistance to the antibiotics 8-fold. Left side. You can see in B, amino acid sequences of the most active mutant colored as in A to show the original regions of the protein, with the point mutations shown in red. The gray regions cannot be ambiguously assigned to one of original genes, this region. you can see the model of the evolved mutant, the protein from the homologous the protein. This chimeric proteins contains portions from the different starting genes.